Hi everyone, welcome back to the channel. This is Dr. Hasid, and today's topic is inheritance patterns or Mendelian genetics. So we have student learning outcomes for this chapter. This is chapter 8 in your orange book. Again, Mendelian genetics. So one of TSLO is basically understanding Mendelian inheritance, how Mendelian genetics works. We will also look at crosses, genetic crosses. We will look at pedigree analysis. We will look at also some non-Mendelian inheritance, and also briefly look at genetic linkage and recombination. So let's get started. Okay, so Mendelian genetics. Okay, so who is Mendel? So Mendel, as you might know already, so he was an Austrian, currently Czech Republic, a scientist. He actually started the very first experimental genetics experiment with basically today what we use and what we call Mendelian genetics. So there are a number of vocabulary in this chapter. So you might want to start with working with vocabulary first, get down some definitions. Heredity means transmission of traits from one generation to the next generation. Genetics is studying heredity is genetics, which is also a technique that's used by plant biologists, animal biologists, other areas everywhere in biology. So Mendel, or Gregor Mendel, did his experiment in 1860s, and he published 2 papers of his results of 8,000 plants that he grew and cross-selected and came up with his Mendelian genetics. So he was the first person to analyze his inheritance patterns and then recording them and then coming up with these principles. So he worked with a plant called garden peas, basically as his model system, and he had so many varieties, so there was a good variation in terms of color, flower color, seed shape, so many other things that actually he could work with. So you see here in a picture of pea plants, and you can see a typical cross pollination and fertilization. You have 2 parents, so you have P1, P2. You actually cross them, and then first generation is called F1. And then if you cross F1 with another F1, you go F2. You can keep going like that. So when Mendel actually crossed purple flower pea plants with a white flower pea plants, you can see all the F1 were purple, and he was curious why they are. And then when he crossed them, he seen something that what is called 3 purple, 1 white, which is one of his ratios called 3-to-1 ratio, right, in genetics. So character in genetics means a feature varies maybe among individuals, and trait is variant of that character. Okay, each of these characters Mendel studied had 2 distinct traits. So, and there is something called hybrid, which is if you cross parent 1 with parent 2, now F1 is also called hybrid, F1 hybrid. Parental generation, which is called P generation. Next generation is F1. Following generation is F2, F3. It goes on like that. Okay. So you can see here is 8,000 plants, and he's so many different variations and traits, and also variations in traits like flower color, purple, white, flower position, axial terminal, seed color, yellow, green, seed shape, round, wrinkle. It goes on like that. Today we have another plant called Arabidopsis, which is our model plant today, among some other model plants. Okay. So when he did crosses, so if there is one gene involved in a genetic situation, he called them monohybrid, just one single gene. It's like the flower color. If there are 2 genes involved in a genetic situation, call them dihybrid. If there are several genes involved, more than 2, then we call this polygenic or quantitative traits. Okay, so there is a formula called phenotype equals genotype time environment. Always remember that genotype is the genetic makeup, which is genetics. This chapter, phenotype is just physical appearance. Environment is everything else, which is non-living, like a diet and climate and light and all that, everything non-living is environment. Okay, so if so there is a word called there is a vocabulary called alleles. Alleles is actually an alternative version of a gene. If a gene, we're talking about eye color, green eye color or blue eye color or brown eye color, these are different alleles of same gene, eye color gene. Okay, there is a term called homozygous. That means two alleles are same. Heterozygous, two alleles are different. Heterozygous sometimes is also called carrier, like in human genetics. Okay, so sometimes we see dominant and recessive relationship. So if in the presence of two alleles, one dominates the other one, it's called dominant. The other one called recessive. Recessive is not necessarily weak. It's just it will show up only if there are two recessive exist in this next to each other. Okay, so there is a Punnett square, which basically means if you, in order to sort this F2, all the offsprings in the F2, you can make a Punnett square. This is how it works. This is a typical basic cross in genetics. If you take parent one, let's say we call it a letter P, big P, big P. In genetics, big P, big letter is dominant, little letter is recessive, always remember. Okay, so big letter is different than little letter in genetics. So parent one is big P, the gamete will be just big P. The parent two is little p, gamete will be little p. If you cross them, F1 will be big P, little p. And if you cross this with another F1, now it's getting a little too many offspring, so we need to actually sort them out in a table called Punnett square. Can put one parent male maybe here, female here, and then I can actually combine them. Then when we combine them, big P, little p, little p, big P, little p, little p. You can see here, anytime we have a big letter P, then that's dominant. So that basically means it's gonna show purple here. Doesn't matter regardless of what actually the other letter is. White color will only show up here if there are two little p's. This is called 3 dominant to 1 white ratio. This is also called 3-to-1 ratio. This is a phenotypical ratio of 3-to-1. So genes that we're talking about here, 1 gene, 2 genes, several genes, they are located on chromosomes in a specific locations. They are called loci or locus or loci. You can see here. The next will be dihybrid cross. A dihybrid cross is basically if you have two genes now you're studying same time. So in this case, gene one is big R, big Y, big Y. Gene two is, gene one is R, gene two is Y, and here are two parents. If you actually do F1 and F2, you will get 16 possible outcomes. Out of 16, and this is the ratio 9, 3, 3, 1. 9 will be yellow round, 3 will be green round, 3 will be yellow wrinkle, and only 1 will be green wrinkle. That means recessive for both genes. This is also called 9-3-3-1 ratio, which means there are two genes, two dominant genes in. Mendel also came up with this law of independent assortment. We also seen this in meiosis, that the independent law of independent assortment, right? So that means alleles actually independently go to the gametes. There is no order, and we don't know which order they're going to. Test cross is if you have an unknown phenotype, and if you want to test that and find out what is the genotype, that unknown genotype, I'm sorry. And so you can cross it with a homozygous recessive of that gene. If it is like big B blank, we don't know. Cross it with little b, and look at the results. You might have two options. You might have all look like one parent. That means then it was big B. And if it is half and half, and we know that that was little b. This is called test cross. Genetics uses also probability, like statistics. So you can actually predict the results by just calculating it, just using probability. Like drawing, basically you can see here a coin. If it is tail, face, we can actually, this is 1.1 and half, I mean half, 1.2% chance, can actually calculate this in each genetic levels. So this was so far Mendelian genetics, and Mendel actually studied plants, but in human we don't really we don't do that. That's not the way human genetics studied. In human genetics, they studied using pedigree analysis, which means you look at the make a tree, family tree. You look at the individual, you go backwards, parents, their grandparents, great grandparents, uncles, aunts, so all this whole family history. That's called pedigree analysis. Some follow like Mendelian genetics, like again dominant recessive 1 gene 2 gene problems. So if like you can see here some examples, dominant traits like freckles, widow's peak, you see here, free earlobe. Recessive will be no freckles, straight hairline, attached earlobe is recessive. This is not very common, the attached earlobe. So you can see it's actually a recessive trait. So recessive traits don't just go and disappear, but they are, of course, not as common as dominant ones. So, and there is something called wild type, which means it's wild type is just natural, what is found in the nature, versus mutant, which is non-wild type. Okay, so and which is mutant. So we see 1 gene 2 gene and pedigree analysis. You can work the pedigree analysis making a tree like this. You can see a square is male and its circles are female, and in its square circles are filled. It's dominant. If they are empty, recessive. You can actually follow each of these traits that you're working on, and then find, predict the next generation. So here are some examples that you should be studying and remembering the names. So these are some recessive disorders like albinism, cystic fibrosis, sickle cell, and here are some dominant ones like Alzheimer, Huntington disease, hypercholesteremia, which are dominant disorders in human, and these are ratios of these each. So here is one example, which is cystic fibrosis, which is a recessive disorder, and it's found in 1 out of 31 Americans. It's called cystic fibrosis. Sometimes we see dominant situations like achondroplasia, which is dwarfism, or Huntington disease. Again, we're just saying, okay, dominant. Genetic testing. So people can go and actually test it for specific genes, and if they carry those genes, if they would, and to find out actually if they are curious about specific genetic situations. Mendel worked on dominance, but dominance today we know that can be various levels of dominance. There is a dominance called incomplete dominance, like we see in this plan here. We see is called snapdragon. If you have a red snapdragon, cross it with white, we see pink. If you cross pink with another pink, now we see 1 red, 2 pink, 1 white. This is 1 to 2 to 1. Total is still 4, but this is called incomplete dominance. And this plan is snapdragon. So, and then there is something called codominance, which is like blood types. like you know about ABO blood types. But if one parent is A, the other parent B, we see also a codominant situation called AB, like you see here. AB is codominant, A and B are so strong and they show up together, which is AB. Okay, but A also still dominate O and B also still dominate O. But A cannot dominate B. A is co-dominant to B type. Sickle cell, there's some information here about sickle cell you can read about, and we will see it in the coming chapters. Polygenic is basically a situation where there are more than 2 genes involved. Okay, so it's called polygenic, right? So you can find the ratio, but it will be very large ratio. Like maybe for 3 genes, it will end up with 64. Like a ratio will look like 27, 9, 9, 9, 9, 9, 3, 3, 3, 3, 1, 1, 1, 1. So it's like, well, you don't need to really memorize the ratio. You don't study polygenic genes like we study 1 gene or 2 gene problems. So they are more studied as a group, and this is their distribution, more look like a distribution like a bell curve. Okay. Examples, eye color is considered a polygenic. A yield of corn or in the field is a polygenic. Because cancer is a polygenic, heart disease probably a polygenic. So because there are many genes, these are all complex genetic situations. So it's much difficult to solve these and change them. Okay, so and next is chromosomal basis of inheritance. Basically, we talked about genes, but there are chromosomes. So, and genes and chromosomes have things in common. They are both in duplicates. There are 2 genes, 2 chromosome sets. And so after Mendel, other scientists work on chromosomes. And then, so they found that there are genes, they might be linked. That means they are on same chromosome. If 2 genes are on same chromosome, they are linked. Best examples, also some of the work done by Thomas Morgan at Columbia University in 1905, basically with fruit flies, like you see this example. And then when he actually studied the fruit flies, realized that there are also specific things going on X chromosome. If something is on X chromosome or Y chromosome, they are different than the other chromosomes. Okay, so in fruit flies. So, and then when he actually did his crossings with the fruit flies, this shows some offsprings didn't look like parents. He called them recombinant. And then he calculated this recombination percentage, or actually based on these recombinant. And so here is an example. This is called linkage map. This looks like a highway, like I-10 here goes by. So if you have 3 genes, G, C, and L, and if you know the distance between, if you know the recombination percentage between them, you can actually place them on a chromosome. Like between G and L is 17%, G and C is 9, C and L is 9.5. It's about 17 total. This is actually converting the combination percentage to physical distance and finding mapping genes on chromosomes. This is called linkage map. So that brings us another things that they studied, which are called sex chromosomes. And if any genes are on X or Y chromosome in human, they are sex-linked genes. And especially X chromosome has a lot of other genes besides. So color blindness, like red-green color blindness, caused by genes which are actually in this X chromosome link. So hemophilia, another sex-linked recessive blood clotting trait. And you can see here, you can follow it, which is which was a major disease. And these are all connected to the evolution, and we can actually make a phylogenetic trees using this information from historical times to today. There is actually a review of this information, and here in this slide you can study So keep working on chapter 8 in Mendelian genetics here from the book. So, and once you study this and study the PowerPoints, make sure actually complete quiz number 6. So this is this chapter, end of this chapter here. Thanks for watching, and please feel free to comment and like. So I will see you in the next video. So happy studying.